Alrighty, welcome everyone. Today we're going to be talking about regulatory circuitry. We talked a lot about sort of how do we put together all these different pieces of the genome. As I mentioned earlier, 93% of disease-associated genetic variants that are causal for disease are in fact not found in protein coding regions. Instead, they're found in non-coding regions. So the question is, where are they? Are they hitting promoter regions, enhancer regions, other regions? And if they are hitting enhancers, what are their target genes? What are their upstream regulators? What are the motifs that are recognized by these regulators? What are the dynamics of these control regions across different cell types? So these are a lot of the questions that we're going to be asking today about regulatory circuitry. So today we're focusing on the foundations at the genomic level. And then on Thursday, we're going to be talking about networks that arise from these circuits and how do we understand biological networks. So again, a lot of stuff on the menu today. We're going to be talking about epigenomic dynamics and how do we learn chromatin states across multiple cell types, building on what we talked about last time to sort of understand how we can use this to understand the dynamics of gene regulation. Number two, we're going to be looking at enhancer gene linking. So basically finding out which enhancers are targeted to what genes. And also, what are the different methods that we can do that we can use to do that. So we're going to be looking at three-dimensional genome conformation. We're going to be looking at correlation-based methods. And we're going to allude a little bit to expression quantitative trait loci, or EQTLs, which are genetic variants that impact nearby gene expression rather than impact phenotypic traits. So these are quantitative traits, namely measuring something quantitative like expression, but they are affecting not something at the organismal level, instead something at the molecular level. So we're going to be talking a lot more about EQTLs at the genetics lectures, which are going to be around lecture 17 or so. Then we're going to look at how do we discover these words, these regulatory motifs. And we're going to look at enrichment, expectation maximization, Gibbs sampling, and also deep learning convolutional neural networks for motif discovery. Then we're going to look at methods for global motif discovery using comparative genomics and for instance identification in the same way. And then if time permits, which it most likely will not, we're going to look at massively parallel reporter assays for discovering these motifs de novo. Everybody excited? Awesome. I certainly am. Let's do it. So here we are. We're basically in the last week of module 1. Then we're going to switch to protein structure, then chemistry, and then electronic health records, and so on and so forth. And we've talked about expression. We've talked about single cell. We've talked about sequential data in human Markov models. We talked about epigenomics. And today we're talking about regulatory genomics, but we're going to be building, of course, on all of the previous stuff. And then next time we're going to be talking about networks. So let's review a little bit hidden Markov models that we talked about last Thursday and how we actually can learn with them. Okay. So again, part of gene regulation is understanding epigenomics, because that's what gives us the regulatory control regions like enhancers and promoters, where the motifs themselves are sitting and where the enhancers are bound by different regulators, linking them to their target genes. So basically, a lot of this circuitry is physically instantiated. There's physical links in the genome of regulators binding DNA and DNA looping around to bind promoters and RNA polymerase binding to create the elongation and the transcription of the RNAs and so on and so forth. Okay, so there's a physical basis of these networks. And last time we talked about the challenge of mapping hundreds of millions of short reads to the genome very, very fast. We looked at the Burris-Wheeler transform implemented initially in Bowtie in bioinformatics that basically starts by adding a letter to growing suffixes of a search string and then narrowing down the beam of searching by playing with these pointers that we talked about. We talked about how we can use this in multiple independent marks to start looking at each of these signals and how we can combine the signals together using hidden Markov models, which is what we're going to be focusing on at the beginning of the lecture today, and how this very powerful paradigm of observed versus hidden can basically be applied with arrows between the hidden states to capture the dependencies between different positions along the genome. And this is central because you're more likely to find, say, an enhancer region near a transcribe region than outside in the middle of a bunch of repressed regions. Is everybody with me here? So these relationships were encoded in the transition probability matrix of hidden Markov models that told us what is the probability that the next state at position I is L, given that the previous state at position I minus 1 was K. And that gives us the K to L transition, for example, enhancer to promoter, promoter to transcribed, etc. And then the emissions were the probability of observing the ith character in our string, and this is the observed string, given that the ith state is K. Okay, so that's the probability of emitting a particular character given our generative model. Everybody with me here so far? And then we looked at the foundations for hidden Markov models. Basically, how do we transition between different states? How do we self-transition to stay in the same state? And that has implications for the length of either, let's say, promoter sequences or background sequences. Why does it have such implications? Because the probability of staying in the same state might be 99%, but it decreases geometrically at every time step. So basically, if you look at blank sequence, then the length distribution that you would expect is actually a geometric length distribution for these states. Is everybody with me here? Now, this is just the transition probabilities. Then there's the emission probabilities that basically tell us that from some states, I'm more likely to emit given characters, for example, ACGT, at equal probability with background, but C and G at higher probability in a promoter state. And then we saw how we can actually parse all of that by saying that the all promoter or all background sequences had some probabilities, but that parsing this as background and then this as promoter and this as background was paying this 0.01 very low probability, therefore paying a penalty of 100, if you wish, for transitioning, to only gain a marginal increase of less than twofold in the emission probability of each of these characters. Okay, so for 4 characters, I would gain, or let's say for 3 characters, I would gain roughly twofold, and for one of them, I would lose roughly twofold, but I would pay 100-fold to get these incremental gains. Everybody with me here? So therefore, that transitioning in and out was actually very unlikely, which is the whole point of hidden Markov models. The fact that you don't want to be very jittery in switching between different states. Instead, you want to have some hysteresis, some kind of damping of these transition probability so that you can kind of leverage nearby positions to infer the probability of a given state, given a given observation at a given time point. Can I get a 5, 4, 3, 2, 1? So how well you're following so far? Awesome, lots of 5s. This is great, some 4s. All right. Then we saw how, yes, we could score every single possible parse. We could score every one of the exponential number of parses. Why exponential? Because at every position, I have 2 choices, either here or here, either here or here, either here or here, to the n length, so 2 to the n, which is, exponential. But I can find the optimal parse using dynamic programming. How? By setting up a variable, the Viterbi variable, that I would compute at every position, assuming that I already knew the maximum score at every previous state. And then the score, the maximum possible score at that position, would be a choice of one of the maximum scores of the previous ones and the transition from that state to this state. So if I have a very high score in the previous state, but I pay a very high transition cost, I will not want that. If I have a very low score and I pay a relatively modest transition cost, I don't want that either. But if I have a high score and a low transition cost, then I'm doing great. Everybody with me here? So that allowed me to now update the next best choice and remember the choice that led to it. Remember the arrow, just like we did for dynamic programming and alignment. So by remembering these arrows, when I arrive at the last column, I choose the maximum one, and then I trace back my choices to restore the entire path and therefore the entire parse. Everybody with me here? Good. And this was the actual equation of the current maximum will be the emission penalty times the maximum of this coupled choice of choosing the best previous one, as long as the transition is not too costly. Okay, so it's the product of the previous maximum and the transition. Any questions so far? 5, 4, 3, 2, 1, who's with me so far? Nice, great. All right. So this was the best single path. So this is the best parse. What's a parse? A parse is an interpretation of the hidden state path that gave the series of observations X. Okay, so there's a hidden state pi, there's observations X, and I infer the best such hidden state. But this hidden state was one particular path, one particular parse. Now, if I care about a given position, the amount of the probability mass that is concentrated in the best possible path, the best possible parse, is actually quite small. There's many paths, an exponential number, that can parse that entire sequence. And there's a huge number of paths that go through a given state at a given position. The maximum likelihood path might be going through, let's say, the hidden state enhancer. But there might be 100 other paths, ranked number 2, 3, 4, 5, all the way to the 101, that basically go through the promoter state at that position. So if I care about the parse at a given position, I could basically say, let me take the best path overall, and then look at what position is that path, that single path, going through in position 523. Or I could say, well, how about I sum over all possible paths going through position through enhancer at position 523, and the sum of all possible paths going through position through state promoter at position 523. Who's with me so far? Yes? Awesome. So basically what I'm doing is that I'm adding up all possible paths going through B and exiting, of course, making it all the way to the end. And then I'm comparing that total probability to the total probability of all possible paths going through P instead at position 523. Good? 5, 4, 3, 2, 1. Who's with me on the question? Perfect. Beautiful. All right. Now the question is, how do we calculate that? Well, I can calculate the total probability mass going all the way to a point, to a state at a given position, by changing this maximum that we had before into a sum. And therefore, I am summing up over all the entire probability of all the possible paths. Why? Because if I have the total probability all the way to here and the total probability all the way to there, I can just add them up and get the total probability all the way to there. By recursion, if I calculate that variable now, I can just sum it up to calculate the next one. Okay? So just by calculating this change here, as long as the previous one was the total probability sum all the way to the previous state, then calculating the next one can just be done by also factoring in the transition probability and the emission probability. And that gives me the total probability mass all the way to the next point. And therefore, by iteration, all the way to this point. By recursion. Everybody with me here? Awesome. Now, that gives me the entire probability mass going all the way to here. But what about the one exiting? Well, this was the forward algorithm that basically calculates the entire probability mass all the way back to a given point. And then the backward algorithm does the same thing, but backwards. Okay, very creatively named. So we basically have the forward algorithm that I'm adding up to that point, and the backward algorithm that I'm adding up to that point, and together, they give me the total probability mass. There's one tiny little caveat that I don't want to pay for that emission probability twice. So the forward algorithm will include the emission within it. The backward algorithm will not include the emission within it. Super simple. Okay. 5, 4, 3, 2, 1. Who's with me so far? Beautiful. Lovely. All right. So then that's the forward algorithm. That's the backward algorithm. And putting them all together, I get the total probability going all the way to, going through this point, summed all the way from here and all the way to the end at every position. And that's what we call posterior decoding, because I'm using the posterior probability. Given that I've observed the entire sequence, that's why we call it posterior, because it's after observing the entire sequence, that posterior probability of being in a given state is given by that total sum probability mass. Good? Question? Oh, good. Awesome. All right. Who feels that they're learning stuff? 5, 4, 3, 2, 1. Yes, learning. Good. Awesome. Beautiful. So, all right. So that gives us a different way to parse. The previous way was the best possible single path. This way gives us the best position at a given point summed over all paths. But now the best position at point 522 might be, I don't know, a repeat element. And it's the best hidden state at position 522. And the best hidden state at position 523 might be an enhancer that's like super active. There might not be a valid transition between, I don't know, a super repressed repeat element and a super active enhancer. That transition might actually be 0 in my model. But that's okay. I'm not looking for the total probability of that path. That path might actually have 0 probability, because that transition might just have a 0 probability. And that's okay. If I wanted the best path with transitions, well, I'll just use Viterbi. If I'm just looking at the best parse based on posterior probabilities, regardless of whether it's a valid path or not, then I can just use posterior decoding. And at every position, I can just choose the maximum a posteriori state, the state that has the maximum posteriori probability. Sounds good? Good. All right. It's a lot of complicated math. You don't need to understand every single piece of it. I love saying a bunch of 5s, and that's great. But this is still complex stuff. So, I just want to tell you that if you're like at the edge of comprehension, that's still fine. It's, it's hard stuff. Everybody with me? Awesome. Okay. And now with this destigmatization of numbers less than 5, let me get a 5, 4, 3, 2, 1 here. Who's with me? Still a bunch of 5s. This is great. Some 4s. This is great. Okay, good. All right. So far, we've basically talked about, I have already my transition probability matrix, A, and my emission probability matrix, E. And I already know the probabilities of emitting every one of the characters from every one of the states and of transitioning from every state to every state. Right? How do we calculate now those matrices? How do we calculate those parameters? That's where machine learning comes in. Okay? Again, machine learning is about getting better with experience, and therefore with more data, I should get better at estimating these parameters. How do we estimate these parameters? How do we find the emission matrix and the transition matrix given first a labeled sequence and then given no labels at all? Okay? Given a labeled sequence is super easy. What do? I basically say how often in my labeled sequence, say GCA, AAT, GC, labeled with background, 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 promoter, 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 background, background. How, I don't know, how do I estimate these variables? How do I estimate the probability of emitting A given P? Super easy. Denominator, given P, 3 of them. Numerator, emit A, 2 of them. Probability, 2 out of 3. Sounds good? So basically, I can basically simply count those probabilities and just say, well, the probability of, emitting an A from P is 2/3. Probability of emitting T from P is 1/3. Probability of emitting, A from B is 1/5. G from B is 2/5, and so on and so forth. 5, 4, 3, 2, 1. Who's with me so far? Great, awesome. This is super trivial. This is just simply counting. What about the transition probabilities? Transition probabilities, the probability of transitioning from a B state to a P state. Well, I was in B state 5 times. I transitioned 1 time, so 1 out of 5. From P to B, I was in P 3 times, transitioned once, 1 out of 3. P to P, I was in P 3 times. I transitioned to P twice. Like I stated P here and here, but not here. So therefore, 2 out of 3. Sounds good? And of course, you don't do that with 10. You do that with like billions. Okay? All right. So then that's the maximum likelihood estimation. Of course, the problem is that sometimes you get zeros. So that's why if you have very small data sets, you want to have some pseudo counts. Super trivial. Now, it becomes much more interesting if I don't have any labels. So basically, somebody comes in and says, great, I want you to annotate promoters and background and enhancers and transcribe regions and repress regions, but I'm not going to tell you anything. Here's just a bunch of data. If they gave us some annotations, we could figure out the parameters, just like we did before. If they give us the parameters at least, we could make some annotations, right? If I have the parameters, I can just simply run my model forward. It's a generative model. So I can generate sequences. So if I have a sequence of ACGT and a probability model, I can just simply find the best sparse for that sequence. Everybody with me here? If I have the parse, I can infer the parameters. And if I have the parameters, I can infer perhaps an even better parse. And now that I have a better parse, maybe I can infer better parameters, and so on and so forth. So basically what we can do is start somewhere with some parameters that might seem reasonable, and then infer the annotation based on those parameters. Which annotation? I can do the Viterbi annotation. You can basically run the Viterbi algorithm with some initial set of parameters, and I'll get some annotation. I'll get the single best path. Or I could run posterior decoding and get a much more fancy annotation. This will be the total probability mass rather than the single best path. Everybody with me here? So I can use that to go from a sequence to an annotation. And when I have the annotation, I can just pretend this was the labels that I was given and simply count the parameters the same way that I would in labeled data. Is everybody with me here? So basically the trick is that I'm going to now go back and forth, and I will estimate let's say with Viterbi training, with in the simple case of Viterbi, I can basically pick some best guess for the model parameters. I will then carry out Viterbi to identify the best possible path. And I will calculate the observed transition and emission parameters according to the Viterbi path and some pseudo counts. And I'll calculate new parameters. And based on the new parameters, I'll recompute Viterbi, find an even better path. And I'll calculate better parameters based on the better path. And, using this annotation, I'll just re-annotate my sequence and so on and so forth. So I can go back and forth between inferring some parameters that best capture the current annotation and then making a slightly better annotation using those parameters. And now that I have a slightly better annotation for slightly better parameters, now that I have slightly better parameters, infer slightly better annotation. So I start out with, say, a blank genome. And then progressively, over time, I realized that, I can annotate this blank genome with nothing, no labels in advance, by realizing that, oh, wait, there seems to be one state that has slightly higher GC and another state that has slightly lower GC. And using the subtle differences in these nucleotide frequencies between these two hidden states, then I will partition my genome into two clusters, one cluster with high GC and one cluster with low GC. This is exactly the same thing that we did with k-means clustering. We started out with nothing. It was unsupervised learning. And then we said, well, let me throw some centers there. And based on the centers now, I use the nearby points to refine the centers, and I eventually converged to some parsing, some assignment of the points into a generative model that was able to just generate these points with the given labels with maximum frequency. Did I know what the labels meant? No. I just found some blue points and some red points. Later, I may have realized that the blue points were, I don't know, single cells from heart, and the blue points were single cell from, I don't know, cardiomyocytes or, endothelial cells, okay? In my single cell profiling of the heart. The same thing we did for single cell of basically first clustering and then labeling. Same thing we did with k-means of first clustering and then figuring out what clusters mean. Same thing we're doing now. We're basically clustering the genome in an unsupervised way into, say, high GC and low GC regions, but now with the additional constraint that the transitions matter, and therefore that there's going to be some sequential homogeneity, consistency, organization in these parts. Everybody with me here? Can I get a 5, 4, 3, 2, 1 as to how well you're following? Yeah? Okay, awesome, great. So that's the key idea of unlabeled data. It's going to be unsupervised learning through an iterative approach that every single time refines the guess based on the parameters. And then based on these parameters, I'm refining the annotation. And based on the new annotation, I'm refining the parameters and so on and so forth. Okay? So that's with Viterbi training. And this was a simple case because I just calculated a new path. And that's easy. But now, that path, as we mentioned, might capture a very small fraction of the total probability mass. So I want to capture all of the probability mass. And that's what Baum-Welch training does. It basically uses, instead of one path, all paths. And just to review this matrix a little bit, we basically saw how we can score one path, how we can score all paths, how we can basically decode one path, and how we can decode all paths, how we can do supervised learning with one path, and now how we can do supervised learning, unsupervised learning with all paths, okay? Supervised learning, if it's annotated, super trivial, I just count. If it's not annotated, unsupervised learning, I just do Viterbi training, find the best path and annotate using that recursive. And now if I want to do unsupervised learning over all possible paths, then I do Baum-Welch training. What is Baum-Welch training? Baum-Welch training basically says, I have some parameters, and I want to know, based on my existing parameters and my existing sequence, how often do I transition from, let's say, promoter to background? Okay. And I want to know from background to promoter, how often do I make that transition? And I also want to know how often do I generate A from a background sequence? But now to do that, what I'm going to have to do is basically say, if I want to count every possible way in which I could emit the A from a background state here, then I'm going to have to sum over all the emissions, all the possible paths, down to ending up at B, and then emitting the character that I have observed. This is the only thing that I have, based on the total parameters. So basically the question is, instead of taking a single path, what I'm now going to do is I'm going to sum over all possible paths ending up at that state, and then of course continuing on to the entire sequence at position I. And then, now that I have that total probability, I just count how many times did it emit A. So instead of having a single parse that basically says, oh, in this best state, I emitted A. I now do all possible parses, each weighted by their probability, using posterior decoding. And from that total probability mass, I say, okay, well, I emitted this, that fraction of the time. Okay? So instead of having a single path, I now have all possible paths down to that position, and then I count that single emission. Everybody with me here? And I can do the same thing here. I can basically use posterior decoding using the forward algorithm all the way down to that position in this state, using the backward algorithm all the way down to that position at that state, and then calculate that transition probability. And that transition probability, from K to L, is simply saying all of the forward vector, that transition from KL, the emission from L, and then the entire backward vector. And this one just simply says the entire forward vector to here, the entire backward vector to there, from there, but backwards, and the subset of states, summing over the subset of states that are actually B in that posterior decoding. Okay? Let me get a 5, 4, 3, 2, 1 here. Let's see 5s. That's pretty impressive. Let's see some 4s. This is great. Okay. Again, this is one of the hardest things in today's lecture. Okay. Good. And then this is simply the derivation, but you don't have to worry about that. It's like for your own exercise. But then the idea is that, that's how we figure out that it's all the way to the forward, all the way from the backward, and then these two transitions for that. Okay. So using now this hidden Markov model capabilities, we can basically go and let it loose on the genome and basically say, hey, what do you find when you scan the entire human genome? And it basically comes back and says, oh, I found that there's a subset of regions that have higher GC content. And then we ask, where are they? And it says, well, you guys check. So we basically search, are they enriched in repeat elements? Are they enriched in enhancers? Are they enriched in transcription start sites, in transcription end sites, in, gene exon transitions, etc. And then we find that, oh, wow, they're enriched in transcription start sites. So then we can label that hidden state that it discovered as probably a promoter associated state. Everybody with me here? So that's exactly what we're going to do with our chromatin states. We're basically going to annotate the genome, partition it into a set of states that have some kind of emissions and some kind of transitions, and then go and actually annotate the states. So then we're going to say, okay, great. Here's what we found. We were able to partition the genome, let's say, into 50 states. And then what we realize is that there's a bunch of states that are associated with the start of genes. There's some other states that are associated with the middle position of genes. And there's some other states that are flanking active genes. And there's some other states that are further away in repressed regions. So we can use this to now start interpreting what is the actual meaning of those states that we discovered de novo. Everybody with me here? So we basically took a bunch of epigenomic data, we parsed it into a hidden Markov model that emits multiple marks at once, and then we take the result of that unsupervised de novo training of learning the best parameters that can partition the genome into states that have similar epigenomic signatures. And when we have that partition, when we have that parsing, we're then able to say, okay, now maybe these promoter states can be annotated as such. Maybe these purple states can be annotated as transcribed. These orange states can be annotated as enhancers, and so on and so forth. Yeah. This is an excellent question. Do we tell the Baum-Welch algorithm how many states we want? We can basically tell it, why don't you try 20? Why don't you try 30? Why don't you try 40? Why don't you try 50? And for every one of those, it's going to say, here's a great answer, where the number is exactly 50. So yes, we have to tell it how many. But then the question is, after the fact, which model do we choose? And that's going to be the subject of the next section. Other questions? All right. So now here's what fraction of the genome was assigned to different states. You can see here that with 1, 2, 3 states, we basically have more than half the genome. I'm going to call this boring. Okay, so more than half the genome is kind of boring when it comes to epigenomics, at least with his marks. And then here, there's like less than 5% of the genome. That's like 12 states to annotate it. I'm going to call this exciting. So the genome, the model is able to basically say that for some small chunks of the genome, it's going to use a lot of expressive power. For the vast majority of the genome, it's going to say, meh. Everybody with me here? This is kind of exciting. This is what the actual parse looks like. So basically, if we take these 50 states and then we parse all 23 pairs of chromosomes and the X and the Y, then what do we see? We see that there's these states here that are only used kind of like in the middle of chromosomes, like maybe near these centromeres. And we see some other states where, you see like a lot of concentration where like a lot of stuff is happening. So this is kind of exciting. It basically says, and you can see that in X and Y, there's like big chunks where, not really much is happening. Okay. Actually, coming to think of it, maybe we should have learned a specific chromatin state model just for X and Y because they may have different epigenomic marks. But anyway, this is kind of exciting. It basically says that we can kind of parse now the genome and assign it to different regions. It also says that different chromosomal territories have different types of activities, which we're going to come back to in a couple of slides when we realize that there's like two compartments. There's an A compartment and a B compartment in the genome. The A compartment is the active compartment, easy to remember, and it sort of sits in the middle of the nucleus. And the B compartment is the repressed compartment, and it sits on the periphery of the nucleus, which we call the nuclear lamina. It's kind of cool. We can kind of discover that, de novo. Everybody with me here? So we've discovered our emission matrix and our transition matrix, and now we want to give them names. We want to call them, this one we're going to call Jill, and this one we're going to call Jack and Robert and Sally and so forth. But much more seriously, we're going to give them names that are meaningful. So for example, we're going to call those promoter states, and we're going to call those transcribed and active, intergenic, and repressed and repetitive. Why? Because when we looked at enrichments of different states for different functional annotations that we already had. I mean, this is happening in 2010. Like, people have spent like decade plus trying to annotate the genome. And what we found at the time, back in 2010, is that these states here that we call promoter states were very strongly enriched in the 2000 nucleotides flanking the transcription start site. Kind of cool. So from the epigenomics alone, we could see that they were enriched here. So that's why we call them promoter states. Not like promoter states are here. It's like, no, we call them promoter states because they're here. Everybody with me here? Then we looked at repetitive states, and then we found that they're enriched in these repeat elements. And basically, if you scan across all of these enrichments, you see that these states here are like hugely enriched for repeat elements. So we're going to call these repeat states. Everybody with me here? And then for these, we found that the association with this lamina, of like the nuclear lamina, the proteins that are coating the membrane of the nucleus, is very strongly enriched in those states. So we're going to call these repressed states. Everybody with me here? So it's kind of cool. We were basically figuring out what the genome means just by doing this. And then we asked, what about annotated genes, transcribed regions? Well, big enrichment for those states here. So we're going to call those transcribed regions, and so on and so forth. Everybody from here? And then we can go a step further. We can basically start like annotating them with much more detail. And this is what we did. And, these are the names that we kind of came up with at the time. And it's kind of exciting. If you look at the different promoter states, they were actually enriched in different functional categories. If you looked at the transcription end state, what we found is that there was one chromatin state that was actually found right next to the transcription end that was peaking there. We found another state that showed 112-fold enrichment for zinc finger proteins. I mean, in the genome, you're kind of excited when you find a 2-fold enrichment or a 5-fold enrichment, but a 100-fold enrichment, I've never seen that. But this 100-fold enrichment was basically saying that we could identify these, and there's this whole paper that basically talks about this CAP1 co-repressor function that basically has the emission matrix in its abstract. So it's like RNA polymerase 2, K9 acetylation, K4 methylation, histone occupancy, trimethyl histone H3K9, H3K36, and histone H4K20. It's kind of cool, right? Like basically, this is not like some obscure paragraph in the paper. This is like the actual abstract of the paper. So we also found that promoter states versus enhancer states had very different transcription factor occupancy, a very different motif occupancy. We found that even 10 kb away from the transcription start site, you could still predict whether something was going to be expressed just from those distant states. And we found very distinct types of repressed elements. So, not all repressed regions are the same. In fact, they associated with different chromosomal bands. And basically, when you stain chromosomes, you see that some regions stain more darkly than others. And those are actually associated with different histomodification marks. Okay. You could also see that these states show distinct methylation, distinct DNA accessibility, distinct transcription, distinct acetylation, and so on and so forth. Okay. This is also very exciting because we can now start annotating the genome. We could basically say, well, there's perhaps a promoter region here flanked by this active intergenic region that overlaps this, high conservation region. And in these, we actually found new protein coding genes. We also found that there were signatures of promoter followed by transcribed in regions that were clearly not protein coding based on evolutionary signatures that we're going to talk about at another lecture. And those regions turned out to be a whole new class of long intergenic non-coding RNAs or link RNAs that were basically, very strongly non-coding based on these codon substitution frequency score that we had developed in some other work, and yet had all of the signatures of polymerase II transcription. So, there's a whole, field studying these link RNAs. We could also find new developmental enhancers using this annotation. And we also found this one epigenomic state that was very enriched in genome-wide association studies. Remember, when the genome was first annotated, people said, oh, let's only focus on the protein coding genes. The rest is just a bunch of repeat regions. Why would we spend all this money sequencing it? And then when we started doing genome-wide association studies, we found that only 7% of the genetic regions associated with disease were in fact within protein coding genes. The others were out there in what people call the genome black, dark matter. And for the first time, we had a flashlight to look at the dark matter. And we found that the flashlight that was painting enhancer regions was in fact co-localizing with single nucleotide polymorphisms that were associated with disease. This is kind of cool. So we had now a way to get at these regions. And we can start asking, how well can I discover transcription start site, transcribe genes, and so on and so forth. Okay. So going back to the question of, should we have used 50 states? Is that enough? Should we have used maybe 100 states or maybe 10 states? So the question now is, how do we select the complexity of our model? And this is a question that you're going to ask all the time in every single type of analysis that you do, whether it's single cell, whether, in the number of cell types, whether it's, for like unsupervised clustering of expression patterns. How many clusters are there? Are there 20? Are there 100 or 200? So you can use all kinds of ways to ask that. One way that you can use is to ask, is there some kind of mark independence that becomes visible when we have more states? And in particular, you can ask, what is the co-occurrence frequency of pairs of marks within a state if I have 2 states or 4 states or 10 states or 100 states? And what we found is that as you increase the number of states, the deviation from expectation decreases. And with 51 states, you're more or less on the diagonal, which basically says that co-occurrence frequencies of pairs of marks within the same state is more or less as you would predict. Whereas with fewer states, you see a lot of deviation from these expectation. And that sort of has some connotations about, oh, well, maybe with 5 states, we're only capturing mark-independent effects, but with 50 states, we're also capturing the mark dependencies. Okay, so that's one way to do it. Another one is you could select the number of states using this nested initialization approach. What does that mean? That means that instead of just saying, oh, I'm going to choose 50 states and I'm just going to run my model, and then whatever 50 states it finds, I'm going to be happy with. Instead, you say, well, why don't I run the model all the way to, I don't know, 80 states? And then I can start pruning down states that are redundant. And that's a very powerful general technique that you should be thinking about. Why is that helpful? Because it basically says, yeah, sure, I can choose 50 states, but will the model randomly fall on the 50 states that are awesome just by chance? What if there's, what if it captures, I don't know, 3 times state 27 and 2 times state 23, and it doesn't ever capture state 48? Are you guys with me? So basically by first learning the larger model and then pruning it down to eliminate the least informative states, or at least the most redundant states, then you have the chance to perhaps also capture state 47 that doesn't always come up, and then to eliminate the 3 versions of state 23. Is everybody with me here? So that's what we basically did. We basically said, okay, let's learn the larger model, let's prune it down greedily, and then just select some arbitrary cutoff based on how many states we can actually make sense of. Were we right? Yeah, maybe not. We'll find out. But the key idea is the following. If you do this random initialization, and then you ask, how often do I recover a given state of this 80-state model? Then you see that this particular state here, you recover it here, and then you lose it, and you recover it again, and then you lose it, and you recover it again, and eventually you recover it consistently. And this other state, you recover it here, but then you lose it there, you recover it again, you lose it, and so on and so forth. But in the nested initialization, what you basically see is that as soon as you get 7 states, you recover that, and then you never lose it again. And, this one state, you recover maybe at, I don't know, 41, and then you never lose it again, and so on and so forth. Is everyone with me here? So basically, the model is much more robust because it doesn't depend on that random initialization capturing every single one of those rare states. Make sense? Again, think of this as a general approach when you're like discovering clusters in your k-means algorithm. You could say, well, yeah, sure, I could run it once and hope I get lucky, or I could run it to many, many more states and then collapse them down and then take the unique states that I've discovered. Yes? Yeah. 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 Great question. So basically here, we don't care what they are. We start completely unbiased. We prune completely unbiased just based on the redundancy of those states. And then in whatever final model, that's where we bring in the biological interpretation. Now, the place where this matters is that we basically say, okay, maybe this state here, I don't recover until much, much later. And I would have to have like, I don't know, 75 states to recover it. And I can't really tell what that state is. So I'm just not going to care about this. But we're not going to arbitrarily say, oh, well, these ones, I have no idea what they mean. So like toss them out of the model. That's the cheating part. That's the sort of overfitting to known biological stories. But basically, the moment where I said, oh, you choose some arbitrary cutoff, that's basically the stuff that we could interpret. So I agree that that's overfitting to known knowledge. And it's possible that state 52 is actually super interesting. And with new data, since we did this work, we could have said, oh, well, let's also keep this one. Does that make sense? Other questions? Okay, awesome. And then you can kind of see here that with, random initialization, you basically very rarely recover the given state. But now with this nested initialization, you can kind of see that, like, you kind of recover the state, boom, and then you kind of keep recovering it, and so on and so forth. Okay. And great. So now this is now learning these states using a single sequence. But if you have multiple cell types, then you can actually learn these chromatin states jointly across multiple cell types, okay? And for example, if you have human vein endothelial cells and keratinocytes and lymphoblastoid cells and, myelinating leukemia and liver carcinoma and human lung fibroblasts and epithelial cells from the mammary and skeletal muscle myoblasts and embryonic stem cells, then you can measure the same marks in the same cells, and you end up with this giant matrix of 81 different chromatin mark tracks, which is the 9 marks times 9 human cell types. The question is, how do you learn a single set of chromatin states? And there's many strategies that you could take. You could basically say, well, I'll ignore the fact that this mark is profiled in each of those, and that each of those is profiled for all 9 marks. And I'm just going to simply say, oh, yeah, okay, no problem. I have 81 tracks. So I'm just going to learn a single giant model by stacking together all of the tracks from all of the cell types. Everybody with me here? So you could do that, and this is actually very helpful. You could learn states that are only appearing in one cell type. At the same time as you're learning that they're enhancers, you're also learning that they're only in stem cells. And that's very helpful if there's a special class of enhancers that only appears in stem cells. Then you'll be able to sort of find, these uniquenesses. Alternatively, what you could do is basically say, well, instead of pretending that I have 81 different marks, namely the number of marks times the cell types, I'm going to pretend that I have, instead of 23 chromosomes, I'm going to pretend that I have, 23 times 9 chromosomes. And therefore that I have like this just giant genome, all annotated with only 9 marks. So this is what we call the stacking approach, where you're basically stacking the marks on top of each other. The other one is what we call the concatenation approach, where you're just like, concatenating the chromosomes back to back. And therefore, you're forcing your model to have the same state definitions in each of the cell types. Because instead of just saying, oh, I have these marks in 23 chromosomes, which are all the HUVEC experiments, you now say, okay, I have 46 chromosomes, 23 from here and 23 from there. Is everybody here? So basically what you're doing now is that you're running the same model on a concatenated genome that captures, a lot more space. And as I mentioned earlier, solution number 2 was basically this stacking of all of them together. Solution number 3 is this concatenation approach of basically pretending that, I have just much more genome. And then solution number 1 is you could say, okay, I'm going to learn independent models for each of the cell types. And then what you can do is cluster the emission matrices of all of these models that you've learned. So I've basically learned, 15-state model in each of the cell types. And then I just simply say, okay, great. This promoter state one, turns out I recovered it a bunch of times, including in the average model. This state I recovered a bunch of times. This state I recovered a bunch of times. This state a bunch of times. This state a bunch of times. Oh, this state, I only found it like in 2 of the cell types. And what is this state? K36 trimethyl, K4 dimethyl, monomethyl. And then, I'm not too familiar with that state. So basically, maybe we should be paying closer attention. And so on and so forth. Is everybody with me here? So basically, you could sort of discover these states a bunch of times independently, or you could jointly discover them by concatenating cell types, or discover a much larger model that simply stacks all of the cell types together. Everybody with me here? Yeah. Yeah, but it's the same thing that we had to do when we concatenated the 23 pairs of chromosomes. So yeah, it's the same thing. Of course, the boundaries, you don't have to sort of think about transitions between them. You basically just eliminate the transitions at the boundaries of chromosomes. But those are tiny, tiny subset of the regions. Even if you concatenate them, it's not going to learn a new type of transition from telomere to telomere. Everybody with me here? Awesome. Now, there's a challenge when this mark is missing in one cell type, and this mark is missing in another cell type, and this mark is missing in another cell type. Then suddenly what do you do? Well, you could actually start imputing the missing marks. You could say, let me just simply use the rest of the marks that are present to infer the missing information here. And this can be very, very powerful if you have, this giant matrix of experiments from some very rare samples, and one of your experiments failed, and you have no sample left to repeat it. So you have to do something, either eliminate that mark from all of the cell types, or try to figure out what would that mark look like if you did imputation. Everybody with me here? Okay, awesome. So this concatenation approach basically allowed us to learn jointly across cell types with a common set of state definitions and with dynamic locations of the same state. So now you can say, I can talk about an enhancer state, say state 4 or state 5, and I can see how that enhancer state turns on and off across different cell types, or how I'm switching from, let's say, this repressed state 12 to this enhancer state 4 between cell types. Everybody with me here? So now you can incorporate dynamics into your model. So here's all of the data for human embryonic stem cells. Here's all of the data from GM12878, from HUVEC, from NHLF. And then what you can see is that you can now learn these dynamic chromatin states across the 9 cell types. And what do you see? You see that this gene here in the embryonic stem cells appears to be in this poised promoter. And indeed, it shows marks of repression, H3K27-trimethyl, and marks of activation, H3K4-Me3 and Me2. By contrast, in these 5 cell types, it appears to be active. You see these promoter marks. You don't see any repression from polycomb. It's gone. And then you see these transcribed marks as well. Everybody with me here? So therefore, you can reason about the gene turning on in those cell types, turning off in those cell types, and then being poised in those cell types. Is that cool? Yes? So just by learning these chromatin states jointly across cell types, you can kind of see the dynamics of the genome coming to life, like turning on, turning off, being poised, and so on and so forth. And that's true for all of these genes nearby. And you can kind of see just how powerful and how clear this approach is. You can kind of see promoter states, transcribed regions. You can see these active intergenic regions. So now if you have a genetic variant that's sitting somewhere here, you can be like, oh, yeah, well, something's happening. Or another one is sitting here, you're like, well, that's probably not functioning. Is everybody with me here? So now you can start annotating the non-coding regions. You can see this beautiful gene, how it's, transcribed, the promoter states. In HUVEC, something's going on. There's more repressed regions here, and so on and so forth. Kind of fun. You can spend all day just studying different parts of the genome. But what's really exciting here is that you can now go a step further. You can start reasoning about this genetic variant that sits here that appears to be active in these cell types is near a gene that appears to be active exactly when that non-coding region becomes active. It's kind of cool, right? So you can start linking together non-coding regions to their target genes. Remember, that was one of our goals earlier, where we basically wanted to predict links on the genome. And you can do that now by looking at the dynamic nature of the genome across a bunch of cell types. So now you can sort of start saying, okay, let's expand this. And we did across, from 11 to 127 epigenomes. And then we did it across 800 epigenomes. And now we're doing it across 2.7 thousand epigenomes, et cetera. So you can basically now start looking at how, for example, promoter regions appear to be stably on, even when the corresponding genes are not actually transcribed. You can see these PAX5 gene is super repressed and it's transcribed from right to left. And you can see here that you see the transcription turns on, and then, you can see promoters and then the transcribed regions, etc. And then very excitingly, you can basically see that this region here appears to be actually associated with the activity of the PAX5 gene. That basically when this region turns on, that entire segment turns on. So you can, again, do this kind of linking, okay? So this is one type of linking. You can basically use correlation-based linking. That basically says whenever these enhancers turn on, these genes also turn on. Everybody with me here? Good. You can use genetics-based linking. That basically says instead of asking for genetic correlation with whether I'm, a case or a control for, let's say, Alzheimer's or schizophrenia, then I can basically look at these genetic variants and see that whenever I have a T, this gene is more highly expressed. So these are genetic links, or EQTLs, where basically these nucleotide variants, these differences in the genetics, have a difference in gene expression as well. Who's with me on the EQTL part? Raise your hands. Awesome, great. So these are two types of links. One of them is functional, based on activity. The other one is genetic, based on EQTL effects. And the third one is based on physical interactions. This is based on the looping of the genome in 3 dimensions, okay? So this is where chromatin conformation capture comes in. So we're going to make a small parenthesis to talk a little bit about Hi-C, or chromatin conformation capture. Here I'm showing you actually one of my favorite regions of the genome that actually contains the FTO gene that is associated with obesity. And then you can see these long-range links with IRX3 and with IRX5 over here, okay? So you can basically see IRX3, IRX5, and, some other very distant links. So what are we looking at here? What we're looking at here is the probability or the frequency with which this region interacts in 3 dimensions, physically interacts, folds onto itself with that region here. How can you do that? You basically take a bowl of spaghetti or noodles, and then you want to know which noodles are near each other, which chromosomes are near each other, which chromosomal segments are near each other. So you're going to take your chromosome noodles, and you're going to chop them up. First, you're going to add some fixation agent to just freeze them in place. Then you're going to chop up the genome in a bunch of places, and then you're going to randomly allow them to rejoin. And most of the time, they'll rejoin with something that's sitting very close to them in the coordinates of the genome. And that's what you can see here on the band. You see that most of these regions are just rejoining randomly after chopping them up with something nearby. But every now and then, they will rejoin with something far, far away, because that far, far away thing was kind of crossing them. And now, after you've chopped them and you religated them, you've basically now connected this position here with that position here. And that basically allows you to put a dot in that off-diagonal entry. Can I get a 5, 4, 3, 2, 1 as to how well you're following on this one? Yes, that's pretty awesome. Good. Any questions so far? Good. So let's dive into Hi-C. So again, the mammalian nucleus, this is just the nucleus. The cell itself is enormous around it. This is just the nucleus. And within it, you see the nucleolus. But you see that there's a lot of compartmentalization, that basically chromosomal domains exist, where basically you have a lot of functional units, that the DNA that sits out here is very different than the DNA that sits in there. And this is, looking at the cell nucleus under the microscope, and you see that there's really 2 different types of chromatin. You basically see the giant nucleolus here in the middle, and you see the cytoplasm out there. You see DNA in here, and then you see this nuclear lamina. This is the boundary of the nucleus itself. And this thing is tiny, and there's 2 meters worth of DNA. And then you sort of, you compact it to go from the DNA to this looping, to these, different levels of the chromatin, and then eventually the condensed chromosome. But what you notice is that there are chromosomal territories. In other words, if you stain chromosome 1, 2, 3, 4, all the way with different chromophores that basically allow you to combinatorially distinguish each chromosome with a unique combination of colors, then you end up with seeing that the different chromosomes are in fact sitting in different domains, that they're not just like all, squished up against each other. And that's where the idea of chromosome conformation capture comes in. That basically you can kind of cut the DNA, let them loose. If they rejoin, you can sequence past the junction. And by sequencing past the junction, you know that these two regions were in fact in physical proximity with each other. And, it started out with chromosome conformation capture, which is like 3C, and then with carbon copy chromosome conformation capture, which is, 5C, and then eventually with high C, that basically says, I'm just going to add a biotinylation mark to every time I make a cut and a religation. And therefore, I'm going to be able to pull down all of the segments that religated that were not identical to each other. Then I'm going to sequence those. And that's sort of going to be, and that has been the technique of choice for, analyzing this three-dimensional chromosomal structure. Okay? So you can basically start doing the sequencing and then saying, aha, using this biotinylation mark, I know that this orange region and this green region that were actually far away from each other came closer to each other. So I'm going to put a dot at that position. Everybody with me here? And then I saw that these other ones became, dots as well. And then I can kind of use that to start filling in all of these off-diagonal entries. And what we realize doing this is that there are territories on the genome. That basically, first of all, every chromosome sits in its own territory. And that within the chromosome, as you start zooming in, you see basically more and more of these territories. Is everybody with me here? And then you can basically ask how many interactions are in cis, namely within the same chromosome, and how many are in trans, namely across different chromosomes. You can also ask within a chromosome, what are these territories and what do they mean? And the main things that we're understanding is that there is, in fact, some kind of checkerboard pattern on the genome that basically allows you to sort of recognize that there are all of these regions. Do you see this checkerboard pattern that basically says that all of these regions interact with, all of these regions, and all of those interact with all of those, but you don't have this sort of, whole block. And this checkerboard pattern basically says that there's really two giant classes of interactions. And in one of them sit basically all of the genes. And in the other one sit all of these giant intergenic regions, okay, to some approximation. So there's some very, very gene-rich regions, and these are sort of sitting in the middle of the nucleus. And there's some gene-poor regions that are sitting on the periphery of the nucleus. Everybody with me here? Good. So there's a lot of techniques for learning about these topologically associated domains, these groups of regions of nucleotides that are sort of sitting together, for understanding what are the mechanisms that bring these regions together. And the growing model that is pretty well accepted now is one of loop extrusion, where you basically have these CTCF factors that are sort of, binding and then sort of pushing between them the DNA, and then creating these loops and therefore these domains where you see a lot more interaction. And, these were initially thought of as boundary elements, and you can still think of them as boundary elements, but basically they're boundaries because they're kind of creating this loop extrusion. Everybody with me here? And then, again, there's like an enormous amount of work that you can do on your project by studying this three-dimensional chromatin structure. All right, so we talked a little bit about how we can, carry out this linking between non-coding regions and their target genes based on correlated activity, based on three-dimensional structure, based on genetic links. And you can also start saying, if I take unbiased clustering of 2.3 million of these regions based on their activity across 127 epigenomes, and I take these vectors of activity and I cluster them together, okay? So for every region of the genome that lights up as an enhancer in any cell type, I can basically get a vector for how enhancer-like is it across all 127 cell types. Is everybody with me here? So basically, that's just building the vector of activity. And the vector is just simply 127 cell types long. And it says how enhancer-like is that element in each of the 127 cell types. Can I get a 5, 4, 3, 2, 1 who's with me here? Awesome, great. So now I can basically simply cluster that 127 long vector and then define groups of enhancers that appear to be turning on together. And that's what we can call modules. So these are enhancer modules. You can see that some modules are extremely cell type specific. Other modules are turning on for all T cells. Whereas there's, others that turn on only in a very specific subtype. And then there's other modules yet that are extremely broadly active. And you can see here that there's a whole group that is active in embryonic stem cells or induced pluripotent stem cells or ES-derived cells. These are the ones here. And then there's others that are more or less active in all of the rest. Everybody with me here? And what's really cool is that if you look at the genes nearby, namely you take all of these enhancers, which are in the same module, and then you ask, when I take all of these enhancers together, they're scattered across the genome, so I can take all of the genes nearby that I've now brought together by clustering, and are these genes showing common gene functions? And indeed, if you look at gene ontology annotations, you see that there's a ton of enrichments for these cell types here, these enhancers here that were active in the cell types in embryonic stem cells and to some degree in brain. And then you see that these are actually enriched in neuronal development. This is kind of cool. This was the cluster that was active in both stem cells development and in brain. Guess what? Neuronal development. Whereas there's another group here, which is only enriched in the brain. And if you ask what they do, you can see, again, two subgroups, one only in brain, the other one in brain and in muscle, smooth muscle, etc. And then these are enriched in learning and memory. So the corresponding genes of these enhancers that are active in brain are associated with learning. And then all of these that are associated with T cells and B cells in various combinations, they're enriched in immune functions. And in here, you basically see this group of enhancers that are active specifically in the heart are enriched indeed in heart functions and so on and so forth. Okay? Who thinks this is kind of cool? So you're basically taking now this ginormous 3.2 billion letters genome and you're partitioning it into little chunks that are active together and you're starting to make sense of their functions. Everybody with me here? And what's really exciting is that you can now say, okay, great. I have now this entire group of enhancers that turns on at the same time. Do they magically turn on at the same time? Or do they perhaps have regulatory motifs within them that cause the regulators that recognize these motifs to go and bind them all scattered across the genome when these regulators turn on? Of course, it's the latter. So we can now start studying the regulatory motifs associated with these chromatin states coming together. And we can ask, what are the motif enrichments of those chromatin modules, of these enhancer modules? And you can also ask, is the corresponding regulator that is known to recognize this motif expressed in the cell types where these chromatin states become active? So we can basically start linking enhancers to target genes based on this correlation. And we can start linking together regulators with these enhancers based on this correlation. Everybody with me here? So let's basically now study the correlation between gene expression and chromatin state activity. So as I mentioned earlier, we can basically look at this H3K27 acetylation signal as a vector of activity, and then look at the expression of the corresponding gene as a vector as well. And then what we're finding is that this enhancer here appears to be correlated in activity with the expression of this gene, enabling us to start predicting those links. And then we can do the same thing for every enhancer and then start asking, what are all of the genes that it's linked to? And for every gene, what are the enhancers that they link to? So now this is this part earlier that I mentioned about gene expression to chromatin. But now I can look at the three-way correlation between the turning on of an enhancer region, the enrichment of the corresponding motifs, and the expression of the corresponding regulator. Okay? Raise your hands if you're with me here. 5, 4, 3, 2, 1. That's awesome. Great. All right. This is where it gets a lot of fun. So we can basically say in the activity of an enhancer across all of these different cell types, what are the corresponding modules? So basically, I can start looking at modules A, B, C, D, E, F, etc. And I can basically find the modules that are active only in stem cells, or only in immune cells, or only in hepatic cells, and so on and so forth, or the broadly active modules. And for every one of these modules, which are now oriented the other way, so basically for every module as rows instead of columns, I can ask, what is the enrichment of that module for motif in red, or a depletion for a motif? Why would a depletion make any sense? So basically in blue here, you can see here a depletion of K562 and GM12878 active regions in the GFI1 motif. So these regions are lacking the motif. What could be going on? What could be going on is that whenever this factor is expressed, it shuts off these regions. The reason why these regions don't have the motif is because if the motif was there, these regions would be shut off. And therefore, the absence of the motif is basically what allows these regions to turn on. Okay? So you can basically now see that we can define signatures of an activator based on the fact that when the motif is enriched and the factor is expressed, those regions are on. You can define the signature of a repressor based on when the factor is expressed, the motif is depleted. Or conversely, when the motif is enriched, the factor is not expressed. Okay? So I can define both activators and repressors. And the activators, I can basically start saying, okay, OCT4 and RFX appear to be activators in embryonic stem cells. This is kind of cool, matches what we know. GATA, PU.1, also known as SPI1, STAT, are enriched in as activators in immune cells. This also makes sense. GFI1, by contrast, appears to be a repressor of immune cells. Everybody with me here? And you can use that to now start, building together the network controlling these regulators, these modules, and the corresponding transcription factors. And you can now start inferring that network. You can start saying, okay, we have, ETS1, PU.1, etc., that activate immune cells. So SPI1 is the same as PU.1. Sorry about that. You have, neuronal factors like RFX4, associated with neurospheres and so on and so forth. You can kind of build a network of regulators. And now when you ask, where are those motifs relative to the center of, transcription start site, then you can see in very high resolution for the transcription start site how the chromatin lies around the transcription start site. But you can do the same thing instead of a transcription start site, which is a fixed region in the genome. You can basically say, let's find all occurrences of a motif, or all occurrences of a conserved motif, or all occurrences of an active motif, and so on and so forth. And then you can look at the chromatin state nearby. And if you look at the hepatic nuclear factor 4, HNF4 motif, you basically see that in HepG2, in hepatic cells, that motif is surrounded by this very active, strong enhancer state 4. But, the repressed state 12, is actually depleted whenever that motif is found. Does that make sense? And then you can kind of ask, how does that look like for, say, the wild type HNF motif or a permuted HNF motif? And you can kind of see here the corresponding luciferase activity in an experimental validation is actually dramatically reduced if you just change this one motif. And of course, motifs don't act in isolation. They act in the context of other motifs. So the expression is not gone completely, but it is dramatically reduced by just changing the single motif. Okay? So that's where we're going to stop for today. We basically talked about epigenome dynamics. We talked about this joint chromatin state learning. We talked about hidden Markov models and how we can do learning de novo on these hidden Markov models. We talked about the two different approaches for learning. One of them is supervised, where we just simply count, and the other one is unsupervised, where we infer and then count. And then this inference can be made based on the best path, or it can be made based on all possible paths. Then we talked about how we can annotate these chromatin states that we learn from the model based on the enrichments of nearby annotations, and we can give them names, we can learn all kinds of things about the genome. Then we talked a little bit about model complexity, about how increasing the number of states captures those pairwise dependencies between different marks, and this nested initialization idea of increasing the number of states and then pruning the low information or the redundant ones, giving you a much more systematic ability to discover these states in a much more robust way. Then we talked about three different ways of learning chromatin states jointly across multiple cell types. One of them is just stacking them up. The other one is concatenating the genomes. And then the third one is learning them independently and aligning the states with each other. Then we talked about how we can link together regions based on their correlated activity, based on genetic information, or based on their physical proximity when you chop up the genome and religate it, using the high-C approach of chromatin conformation capture, but now applied to the whole genome. And then we saw how we can use correlated activity patterns, both to discover targets of enhancers based on the corresponding gene expression nearby, but also to discover repressors and activators based on the relationship between the enhancer state turning on or off, the promoter, the motif that is enriched within that enhancer state or depleted, and the corresponding transacting regulator that is known to recognize that motif and whether it is active or not, and how we can use that to discover these regulatory networks. So who feels that they've learned stuff today? Awesome. So we'll continue on the theme of networks on Thursday, and looking forward to seeing you then.